In this video, we're going to learn about fermentation, which is another term for anaerobic respiration, so respiration that does not involve oxygen. After glycolysis, pyruvate has two choices. It can enter the mitochondrion, where aerobic respiration will occur, so this will happen if oxygen is present. And then the other option is anaerobic respiration, which will happen in the cytoplasm. And depending on the type of organism it is, it could be lactic acid fermentation or ethanol fermentation. Fermentation must occur because if there is no oxygen, the cell would run out of NAD+, which is needed for glycolysis. And there needs to be a way to convert NADH back to NAD+, so glycolysis can continue. Fermentation definitely does not produce a lot of ATP energy, but it is a solution for when there is no oxygen, so at least some energy can be produced through glycolysis. The first type of fermentation is ethanol fermentation, and you would observe this type of fermentation in yeast cells and bacteria. And it takes place in the cytoplasm. And the molecules that enter the pathway are pyruvate and NADH. And then what comes out of the pathway is ethanol, carbon dioxide, and NAD+. So the main idea is to oxidize NADH back to NAD+, so glycolysis can continue. However, this reaction is not reversible because of the production of carbon dioxide. So the carbon dioxide will escape, and then you end up with a 2-carbon molecule, which is ethanol. So this process is non-reversible. So this is a representation of ethanol fermentation. So you start off with your pyruvate, which is a 3-carbon molecule, and NADH gets oxidized to NAD+, and at the same time, a carbon dioxide molecule is broken off the pyruvate molecule, and the atoms get rearranged to form ethanol. So as you can see, you lose a carbon dioxide and end up with a 2-carbon molecule. So you can never go back to pyruvate. And this is quite handy though, because some organisms use this pathway and we use it for food production, so producing ethanol. But also when we make bread, yeast will convert pyruvate to ethanol and produce some carbon dioxide, and it's the carbon dioxide bubbles that cause the bread to rise, and the ethanol would bake off when you're baking the bread. Lactic acid fermentation occurs in our cells, so this also takes place in the cytoplasm of the cells, and the molecules that go in would be pyruvate and NADH, just like with the other one, but the outputs are different. So this time you end up with lactic acid and still NAD+. So the main idea is to oxidize NADH to NAD+, so glycolysis can continue, but this process is reversible. So once oxygen becomes available, another reaction takes place to convert lactic acid back to pyruvate, and then aerobic respiration will take place. So this is a representation of lactic acid fermentation. So you have pyruvate, a 3-carbon molecule. We oxidize NADH to form NAD+, and then you rearrange the atoms and form lactic acid. So again, this one is reversible because we haven't lost one of the carbon molecules or carbon atoms, so you end up being able to reverse this when oxygen is present.